Okay, Karen, this is for you. So let's see. Antibody dependent enhancement. I don't even know what that means. Of SARS-CoV-2 infection in recovered patients. Studies based on cellular and structural biology analysis. A bunch of Asian people in the authors there. I'm just going to read the abstract. Antibody-dependent enhancement has been reported in several virus infections, including dengue, fever, SARS, and MERS, to study whether ADE is involved in COVID infections. In vitro pseudotyped SARS-CoV-2 entry into Raji cells 562 K562 cells and primary B cells mediated by plasma from recovered patients were employed as models. Let's see here. COVID-19 infections. In vitro pseudotyped SARS-CoV-2 entry into Raji cells, K562 cells, and primary B cells mediated by plasma. So they use plasma from recovered patients. Okay. The enhancement of SARS-CoV-2 entry into cells. So this is weird because when they say they're putting, okay, they put pseudotype SARS-CoV-2. Wait, in vitro pseudotype SARS-CoV-2 entry. Is the entry pseudotyped or is the SARS-CoV-2 pseudotyped? The enhancement of SARS-CoV-2 entry into cells. When they say they put SARS-CoV-2 in cells, like what did they put into the cells? Because apparently they didn't isolate and purify SARS-CoV-2. So, was more commonly detected. The enhancement of SARS-CoV-2 entry into cells was more commonly detected in plasma from severely affected elderly patients with high titers of SARS-CoV-2 spike protein-specific antibodies. Cellular entry was mediated via the engagement of whatever that is, receptor, through virus cell membrane fusion, but not by endocytosis. Peptide array scanning analysis showed that antibodies, which promote SARS-CoV-2 infection, targeted the variable regions of RBD domain. To further characterize the association between the spike-specific antibody and ADE, an RBD-specific monoclonal antibody was isolated from a recovered patient, which potently inhibited SARS-CoV-2 infection of ACE2 expressing cells and also mediated ADE in Raji cells. RBD-specific monoclonal antibody was isolated from a recovered patient, which potently inhibited SARS-CoV-2 infection. I mean, I would question how are they saying that they tried to cause a SARS-CoV-2 infection if they did not isolate and purify a virus? Okay. Site-directed mutagenesis, the spike RBD domain, reduced the neutralization activity of 7F3 but did not abolish its binding to the RBD domain. Structural analysis using cryo-electron microscopy revealed that 7F3 binds to spike proteins at a shift-angled pattern with 1 up and 2 down RBDs, resulting in partial overlapping with the receptor binding motif, while a neutralizing monoclonal antibody that lacked ADE activity binds to spike proteins with 3 up RBDs, resulting in complete overlapping with RBM. Our result revealed that ADE mediated by SARS-CoV-2 spike-specific antibodies could result from binding to the receptor in slightly different pattern from antibodies mediating neutralizations. Studies on ADE using antibodies from recovered patients via cell biology and structural biology technology could be of use for developing novel therapeutic and preventative measures for the control of this infection. Patents about the monoclonal antibodies 7F3 and 4L12 in this study are pending. This work was supported by the National Natural Science Foundation of China. So they're trying to say, hey, let's look in people's bodies who have had this expression, and let's try to put it in other people's bodies to try to suppress that action from happening in their body. First off, I don't understand where they're getting the SARS-CoV-2. Let's see here. To study whether ADE is involved in COVID-19 infections. In vitro pseudotyped SARS-CoV-2 entry into Raji cells. So, I mean, yeah, if anybody wants to comment on what they think this all means. The enhancement of SARS-CoV-2 entry into cells was more commonly detected in plasma from severely affected elderly patients with high titers of SARS-CoV-2 spike protein-specific antibodies. I mean, yeah, this is all I'm getting is that they're saying they want to take an expression, or not the expression, they want to take the aftermath of whatever's in the person's blood from after an expression and put it into something else to try to prevent that expression. But if the problem is the expression isn't caused, the expression isn't caused by the expression, and if the expression is caused by other factors, then I would say that it could be quite dangerous to try to prevent someone's body from having an expression, especially if the expression is the healing. So the expression is the body trying to remove waste and damaged particles, from what I've learned. So if you're trying to prevent somebody from expressing, that means you're going to kill them, because the body has to express. That's the body's way of showing that it's going into a healing mode and a repairing mode. So when you say you don't want the body to go into repair, then that's going to cause worse problems. So it's hard to read things like this because their foundation for their study is that a particle comes and tries to attack you, and you're trying to prepare the body like the Berlin Wall or something to prepare for the attack of the microorganism entering your body. Really, there are attacks on the body. There are like environmental attacks on the body and the things that we put into our body. So yeah, reading things like this is hard because everything is like upside down and inside out. So, and it's hard to read through their fancy terminology, but yeah, as far as it's saying that, let's see, in vitro pseudotyped SARS-CoV-2 entry. So I don't know if they're saying that the entry is pseudotyped or the SARS-CoV-2 is pseudotyped. Pseudo means like fake, doesn't it? Or something? Or not, not natural, not the real thing. Pseudotyped SARS-CoV-2. So yeah, I don't know. Anyone else know?